Hi friends, welcome back. So let's continue the photosynthesis part 2. So friends, if you quickly recall in the yesterday class, we understand what is photosynthesis. We read where does the photosynthesis occur, that is the chloroplast, and we understand the outer and inner structure of the chloroplast. Then we started studying about the light reactions. I want every one of you to remember the ultimate goal of the light reactions, because still we are continuing the light reaction. It's a very broad concept we are understanding step by step. What is the ultimate goal of light reactions? Production of ATP, production of the NADPH. Later we understand where does the light reactions are taking place. Where does the light reactions are taking place? Thylakoid lamellae or the thylakoid membrane. And friends, why the light reactions are taking place only on the thylakoid membrane? As we discussed that all the photosystem, ATP synthase enzyme complex, all the electron carriers, all are present on the thylakoid lamellae or on the membrane. That's why light reactions are taking place on the thylakoid lamellae only. So now what are the various type of photosynthetic pigments? We discussed principal pigments, accessory pigments. Later I have explained you clearly about the structure, that is chlorophyll, carotenoid, bilin pigments. If you quickly recall, I have told you that chlorophyll consists of a porphyrin head. Please kindly don't forget this, friends. And it consists of what? Phytol chain. I have explained you exactly what is porphyrin head and what is phytol chain. And what we discussed about the carotenoid, I have told you carotenoid is a isoprene unit, is it? And friends, what we understand by the bilin pigment, I have told you that bilin pigment, it is a linear tetrapyrrole, is it? So friends, we exactly discussed about three pigments, and we have stopped exactly at this particular slide. Okay, so friends, please try to focus from this particular slide onwards. Based on the absorption of light, there were two spectra were derived. What do we call absorption spectra, then followed by the action spectra. Friends, what is the difference between the absorption spectra and action spectra? Once again, I want every one of you listen to this carefully. Absorption spectra means at which particular wavelength the particular pigment absorbs the light. That is what we called absorption spectra. And one very important point every one of you has to remember, in the last class also I stressed this point: whatever the light absorbed by the pigment, it never ever appear in that color, but whatever the light or whatever the color reflected by the pigment, that appears in that particular color. If you look into the graph clearly, on the x-axis we have taken wavelength of light, on the y-axis we have taken amount of the light absorbed. So here in the graph I have given three pigments: chlorophyll A, chlorophyll B, then friends followed by the carotenoids, is it? Now, every one of you try to understand carefully. Chlorophyll A, see here the peak is very high till 500 nanometer. So, which indicates that chlorophyll A absorb dark blue color, light blue color, but it never ever absorb the green color. So, what I told you, whatever the color reflected by the pigment, it appears in that color. So chlorophyll reflecting the green light, so it appears to be green in color. Now you take an example, carotenoid. So carotenoid absorb dark blue light, light blue light, green light, but it never absorb yellow light, it never absorb orange light, it never absorb red light. If you quickly recall, tomato is red in color. Why, friends? It consists of lycopene. What is lycopene? Carotenoid. What the lycopene is doing? Reflecting the red color. That's why tomato is in red color. And if you look into the carrot consists of what? Beta carotene. Beta carotene is a type of carotenoid. What light? It basically reflects orange light. That's why carrot appears to be orange in color. Always keep that in your mind. Now action spectra. See friends, Once again, keep it in your mind. In the absorption spectra, we have given importance to what pigment, what I have defined, at which particular wavelength the particular pigment absorbs the light. So everything with respect to the pigment. And friends, please kindly don't confuse here. Action spectra basically explains us at which particular wavelength, once again understand, at which particular wavelength the particular process will occur maximum. What friends? At which particular wavelength the particular process will be maximum. That is what actually action spectra. Friends, you can take any process. Let's take an example of photosynthesis process only. Friends, at which particular wavelength the photosynthesis will occur at a maximum rate? Of course, friends, if you look into the graph, 600 to 700 nanometer is the most suitable wavelength for the process of photosynthesis to occur. That is what do we call action spectra, the wavelength at which the process occur at a maximum rate. Friends, still we are in the light reaction, so it's a very broad concept. Okay, now what does this particular light harvesting complex means? First of all, where the light harvesting complex is present, everyone look into the given picture. Friends, this is what? It's a thylakoid membrane. On the thylakoid membrane, what is present, friends? Photosystems are present. Inside the photosystem, what are present? LHC molecules are present. In a simple way, suppose example, this is a thylakoid membrane. On the thylakoid membrane, what are present? Photosystems are present. Inside the photosystem, what are present? LHCs are present. Light harvesting complexes are present. Okay, absolutely fine. Friends, what does every light harvesting system is made up of? This you have to remember for the exam. Every light harvesting complex actually made up of two components, which is very important. You have to remember for the exam. It consists of core pigment and it consists of antenna pigments. In the yesterday class, I told to you friends, antenna pigments we can also call it as a accessory pigment. Core pigment is made up of what? This is an important point. Core pigment, whether it is a photosystem 1 or whether it is a photosystem 2, doesn't matter. The core pigment is exclusively made up of chlorophyll A only. Then antenna pigment or accessory pigments are made up of what, friends? They are made up of chlorophyll B, they are made up of xanthophyll, they are made up of carotenes. Please keep that in your mind. So every LHC present in the photosystem consists of two major components: core pigment, which is exclusively made up of chlorophyll A; antenna pigments consist of a B, xanthophyll and carotene. Now let me ask you one interesting question. Why we have given the name photosystem 1 and photosystem 2? Most of the students will say that based on the absorption of light. No, not exactly. See friends, it's a simple. We have given the name photosystem 1 and 2 based on their discovery. That's all. Photosystem 1 we have given name 1 because it is first discovered. 2 second discovered. Now everyone listen. What I told you, what is this particular core pigment? Core pigment, whether it is a photosystem 2 or photosystem 1, the core pigment is always chlorophyll A only. And then why we are calling P680? And friends, why we are calling P700? The point is here, what is the major core pigment? Chlorophyll A. So friends, chlorophyll A at a 680 nanometer, it can absorb maximum amount of sunlight. That's why we have given the name P680. Now if you look into this, The chlorophyll A, which is present in the photosystem 1, it can absorb the maximum amount of light at 700 nanometer. That's why, friends, we will be calling it as a P700. Don't forget that. Friends, once again, I hope every one of you remember the goal of our light reaction. What is the goal of our light reactions? I told you the goal of light reaction is production of ATP and NADPH. Please kindly don't forget this point until and unless we complete this particular photosynthesis topic. Now, if you really understand this particular slide, the light reactions concept will be little very clear. Now our goal is produce these two. How we can produce? By the resonance transfer of energy. Focus every one of you very clearly, friends. See, you are able to see now all this light color. What is this light color we called? Antenna pigments. We can also call it as what? Accessory pigments. What they made up of, we discussed. They made up of chlorophyll B. They made up of carotenoids, and also what, my friends, they also made up of the xanthophyll. That's what we said, right? And these are all antenna pigments. You are able to see, right? This dark green color, that is reaction center. My friends, what we understand, reaction center made up of chlorophyll A. I want every one of you pay attention towards this particular slide. And here you are able to see, right, purple color. What is that? It is electron acceptor, primary electron acceptor, then followed by several series of electron acceptors are present. Now friends, try to understand the concept. What all the antenna pigments will do? All antenna pigments absorb the light and they transfer the energy to where, friends? They are all going to transfer the energy to the reaction center. Where, my friends? To the reaction center. Now everyone understand, once they pass the energy to the reaction center get excited. What happens? Because upon continuous absorption of the energy, reaction center get excited. Once reaction center get excited, immediately what is going to happen? It release an excited electron. Friends, now this is the point. Now, who is going to accept? In the next slide, I will explain you. Who is going to accept this excited electron? This excited electron accepted by several electron acceptor. Now here comes a unique point. Whenever electron is moving from one electron acceptor to the another electron acceptor, it loses certain energy. What, my friends? It loses certain energy. That energy we can use for the production of ATP. So friends, once again remember, in the next slide it will be very clear to you. If you miss this slide, you cannot understand how exactly we are going to produce the ATP in the light reaction. These are all light green color ones are antenna pigments. The dark green color one is the reaction center. The purple color one is the primary electron acceptor, and so on. When all the antenna pigment absorb the light and pass the electron to the reaction center get excited. The excited reaction center release the excited electron. That electron accepted by several electron acceptor. We will see what are those. When the electron is moving from one electron acceptor to another electron acceptor, there is a loss of certain energy. That energy only we used to produce ATP. Now here you are able to see, right? See here, energy is passing from one antenna pigment to another, another to the another, another to the another. That is only we called resonance transfer of energy. Now I will explain you little more elaborately. Focus. Now we are going to fulfill. What we are going to fulfill? We are going to fulfill the goal of our light reaction in ATP and NADPH. First, let's try to understand about the ATP. Okay, so friends, everyone try to understand what I am talking. Photophosphorylation. what is the photophosphorylation? Photo light, phosphorylation, production of ATP. So friends, the point is here, how can we produce the ATP? We can join ADP with PI. Then there is a formation of ATP. We all commonly know that. But friends, do you think joining of ADP with PI is that much easy process? Not at all. If I want to join ADP with PI, what do I require? Of course, my friends, I require energy. From where I am getting that energy? Just now I told you, is it? Whenever what I have explained to all of you in this particular slide, whenever electron is moving from one electron acceptor to the another electron acceptor, there is a loss of energy. I told you, friends, that energy only we will be using to produce ATP upon joining ADP with PI. Friends, actually that energy basically comes from where? From the sun, is it? That's why friends, we will be calling it as photophosphorylation, the production of ATP by joining the ADP with the PI by using the energy of sunlight. There are two type of phosphorylations are there. Photophosphorylations are there. We called cyclic photophosphorylation, non-cyclic photophosphorylation. Friends, and I want you people to remember whatever the points that I am going to cover now. Name itself very clearly indicating cyclic phosphorylation, which means the movement of electrons are completely cyclic manner. That is what do we call cyclic phosphorylation. Unique point you people has to remember, it never takes place in the plant. First point, it never ever takes place in the higher plants. Then where it will occur? It will occur in the lower plants like algae, and it occur in the certain bacteria also. And once again, a very unique point which is used to ask in the exam, how many photosystems are involved here? Friends, in case of a cyclic photophosphorylation, only one photosystem is involved, that is especially photosystem 1. But friends, when you look into the non-cyclic photophosphorylation, it is exclusively takes place in the higher plants. It never ever takes place in the bacteria. And friends, another unique point, the non-cyclic photophosphorylation consists of two photosystem. It consists of photosystem 1 also, and friends, it consists of photosystem 2 also. Please kindly do not forget that particular statement at all. Now friends, listen my words carefully. This electron scheme they used to ask in the exam more oftenly. I'm going to solve some questions once we finish the light reactions of photosynthesis. Immediately we will solve. Everyone pay attention. Why did I say cyclic photophosphorylation? I called it as a cyclic photophosphorylation because the electrons are moving in the cyclic region. In a simple way, you only look into the point, the electron started from here. Once again, the electron reached the same origin. That's why we called cyclic photophosphorylation. And what we have to remember for the exam point of view, what are the type of electron acceptor? So who is the first? See here, these are all antenna pigments. Antenna or accessory pigment, which consist of chlorophyll B, carotenoid, xanthophyll. What's happening friends? The antenna pigments absorb the light and transfer the energy to what? Reaction center. What happens to the reaction center? What happens to the reaction center? Reaction center get excited. Correct na? Reaction center get excited. Upon excitation of the reaction center, immediately what is going to happen? This electron is accepted by several electron acceptor. Friends, Who is the primary electron acceptor? This point you have to remember for the exam point of view without fail, because we used to get these questions in the schematic presentation. I'll show you the questions. The first electron acceptor is ferredoxin. Next one is what? PQ, plastoquinone. There are two type of quinones are there, plastoquinone A and plastoquinone B. Then followed by cytochrome B6F complex. Then followed by PC, plastocyanin. My friends, you only try to understand what I am talking. From the plastocyanin, where does the electron move, friends? Once again, electron reached origin immediately. Electron reached origin immediately. That's why we call cyclic photophosphorylation. Now listen, whenever electron is moving from one electron acceptor to the another electron acceptor, it lost certain energy. It lost certain energy. That energy can be used to join ADP with PI. Hence, there is a formation of ATP. Now, you will get a doubt. Sir, from where this ADP and PI came from? Of course, they are all present in the cell. ADP means what? Sugar, base, phosphate group. They are all commonly present. Wait, friends. They are all commonly present inside only, inside the cell. Hence, there is a formation of ATP. A simple step. Now, my simple question to all of you is that think and answer. My question is, do we really fulfilled the goal of light reactions by using cyclic photophosphorylation or not? Once again, my question, do we fulfilled our goal of light reactions by using cyclic photophosphorylation or not? The answer is no. Why? We have produced only ATP. We did not produce what, my friends? NADPH. This is the statement they use to ask more commonly in the exam. So listen, there is only one photosystem is involved here. Then only ATP is produced. Photolysis of water cannot take place. Photolysis means breakdown of water cannot take place. Then primary electron acceptor. Who is the primary electron acceptor? Ferredoxin. Why I have written these points? Because if you compare what to what, if you compare the cyclic to non-cyclic, the concept will become easy and you can remember very easily. Now listen. Non-cyclic photophosphorylation, we can also call it as a Z scheme. We can also call it zigzag scheme. Friends, what I told you in the cyclic photophosphorylation, there is a involvement of only one photosystem. But if you look into the non-cyclic photophosphorylation, there are two photosystems are there. You are able to see the picture clearly. P680 photosystem 2, P700 photosystem 1. Now you only try to look into this. This is how the electrons are moving. Now friends, if you look into the scheme of electron flow, it look like a sleeping jet. Correct na? That's why this non-cyclic photophosphorylation we can call the Z scheme or zigzag scheme. Friends, don't get confused. Okay, now I will explain elaboratively, just focus. Since two photosystems are involved, right? Now first let's look one by one. Let's take the first one, P680. Friends, P680 also absorb the light. Upon absorbing the light, where does it send the electron? It sends the electron to what? Reaction center. We all commonly know, right? Reaction center consists of what? Chlorophyll A. And who is the primary electron acceptor? Let me write here. What are the electron acceptor in the cyclic photophosphorylation? I don't want you to compare. For your easy remembrance, I am explaining that. Why you should not compare the electron scheme? The reason, the cyclic photophosphorylation is taking place where? In the bacteria. And non-cyclic photophosphorylation is exclusively taking place in the higher plants. That's why you should not compare the electron scheme. You can compare the differences of cyclic and non-cyclic anyway. So in case of cyclic, who is the first electron acceptor we discussed? Ferredoxin. Later, plastoquinone A, plastoquinone B, later cytochrome B6F complex, then followed by the plastocyanin. Correct na? Now you only try to understand the point very clearly, friends. See, the first all the antenna pigment absorb the light, send the energy to the reaction center. Reaction center get excited. Excited reaction center give the electron. Now who is going to accept the electron? The first electron acceptor is pheophytin. Here is what in the cyclic? Ferredoxin. Here pheophytin. Next quinone A, quinone B. Here also quinone A, quinone B. Next cytochrome B6F. Here also cytochrome B6F. Next plastocyanin. Here also plastocyanin. But friends, if you recall what happens in the cyclic photophosphorylation, from the plastocyanin the electrons starts moving from where to where? It is moving once again back to the origin. From wherever it started, it came back. But friends, do you think the plastocyanin start moving back to the origin? No, the plastocyanin starts moving to the next photosystem. What is that? P700. So from P700, where the electron moved to the reaction center. Now everyone has to keep it in your mind. Now who is the first electron acceptor here? A naught. Don't get confused. A naught nothing but modified chlorophyll A. That is only we called A naught. Then A1 we called phylloquinone or a phylloquinone. Then there is a series of iron sulfur protein. We called iron sulfur X, iron sulfur A, iron sulfur B, FD ferredoxin, FNR ferredoxin NADP reductase. What was that? Ferredoxin NADP reductase, then followed by NADP plus, then there is a production of NADPH. Simple. Let me summarize you. First what friends? P680 transfer the energy to the reaction center. Reaction center get excited. Excited reaction center release the electron. That particular electron is accepted by pheophytin. Now that particular pheophytin send the electrons to quinone A, quinone B, cytochrome B6F complex, plastocyanin, P700. P700 then send to A0, A1, iron sulfur X, iron sulfur A, iron sulfur B, ferredoxin, FNR. FNR is what? Ferredoxin NADP reductase followed by NADP plus and NADPH. Now let me ask you a very simple question. Which is the electron donor? My question is, which is the electron donor for the P700? Who is going to donate the electron for the P700? Plastocyanin. Now second question, which is the first, listen the question, which is the first electron acceptor that accept the electron from P700? What is the answer? A0. A0 is the one which is going to accept the electron from the P700. Now next and very important question, most of the students will give a wrong answer. The question is, listen my question carefully, which is the last electron acceptor that is involved in the production of NADPH? Once again listen the question, which is the last electron acceptor that is involved in the production of NADPH? Friends, the last electron acceptor is NADP plus. Most of the students will say either FNR or FD. No, that's not the correct. NADP plus is the final electron acceptor that is involved in the NADPH formation. Please kindly do not forget this particular statement under any circumstances. Now here come very important point. Most of the people will think that two photo systems will work step by step, which is absolutely a wrong statement. What friends? Two photo system never ever occur step by step. Two photo system works simultaneously. This is a very crucial point. What do you mean by that? Which means everyone understand P700 is also absorbing the light, no doubt about it. P680 also absorbing the light, no doubt about it. Now friends, the point is very clear. Now, when the P700 absorb the light, send the electron to the reaction center, followed by different electron acceptors are accepting the electron. Okay. Now the point is very clear. Now, if the P700 transfer the electron, this is a deficient of electron. Yes or no? Now, who is going to replace this deficient electron? This deficient electron can be replaced by plastocyanin. Never forget that. Plastocyanin is replace the deficient electron for the P700. No problem at all. At the same time, P680 also absorb the light. It also send the electron to the reaction center. Now friends, P680 also deficient of electron. Yes or no? Yes, of course. Now, once again you have to understand. Now it is also deficient of electron. Then who will replace this electron? That's the point. Friends, the electron deficient for the P680 is replaced by water molecule. What do we call it as? We call it as a photolysis of water. What friends? Photolysis of water. Anyway, when the one water molecule break down, what we are going to get? Everyone listen. If you understand this particular point, then only we can understand how the ATP can be synthesized. Everyone listen. When one water molecule break down, what we are going to get? We are going to get two molecules of proton plus two molecules of electron plus half molecule of oxygen. Never ever forget this particular point. So who is going to take the electron? As P680 is a needed electron, then P680 will take this particular electron. What is the fate of proton? This is important. Friends, protons released into the lumen. Protons released into where, friends? Lumen. Now, what is the fate of oxygen? This is the oxygen which we use for breathing. So, this oxygen will enter into the cellular respiration. So when one water molecule break down, we get two molecules of proton, two molecules of electron and a half molecule of oxygen. This is the oxygen which we use for breathing, and we will get two proton released into the lumen, electron can be accepted by P680. What kind of questions we can expect here? A simple questions. Everyone focus. How many number of oxygen? See, the question is like this. How many number of oxygen molecules? How many number of protons? How many number of electrons are released when 100 water molecules are breakdown? 100 water molecules are breakdown. Friends, I have told you very clearly. When one water molecule breakdown, we get a two proton, two electron. Don't think that it's a full oxygen, half oxygen. So for 50 water molecule, for 100 water molecule, how many oxygen we will get? 50 oxygen we will get. 200 protons we will get. And friends, 200 electrons we will get. Do not forget this particular point at all. 100 water molecule. So one water molecule, two proton, two electron, half oxygen. If you remember this, how much number they may ask, you can answer it very easily. Now here comes a unique question. Most of the people will give wrong if you don't understand it clearly. My question is, please kindly don't compare with the cyclic photophosphorylation. As I told to all of you, it is taking place in the bacteria. Now my question is very, very, very clear. Non-cyclic photophosphorylation. My question is, do we really fulfilled the goal of light reactions by using non-cyclic photophosphorylation or not? Once again, do we really fulfilled the goal of light reaction by using the non-cyclic photophosphorylation or not? Friends, we will fulfill, no doubt about it. But in this particular slide, you can observe, we produced so far NADPH only, friends. We did not produce the ATP yet. We did not produce, right? We produced only NADPH. But what I want you to know here, we will compulsory produce the ATP in the non-cyclic photophosphorylation. There is a no doubt about it. Let me explain you how. This is a very, 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 very important point or information which you have to remember for the exam point of view. ATP synthesis. This ATP synthesis, everyone remember, the ATP can be synthesized by a force which we called proton motive force. Proton motive force. Now don't confuse, I will explain in a simple way. Listen to me carefully. P680, these are all what? Electron acceptor, PS1, ATP synthase enzyme. I have told to all of you, listen to me carefully. I told to all of you very clearly, electrons are moving from one photosystem to the another photosystem via several electron acceptor. Listen my words. Electrons are moving from one photosystem to the another photosystem via several electron acceptor. Yes or no? Of course yes. Now every one of you has to remember one more point. Listen, now let me explain you how we can synthesize ATP. So see here everyone, P680, you remember very clearly, it sent the electron to first whom? Pheophytin. I hope everyone remember. Pheophytin is the primary electron acceptor. Listen friends, I'll explain twice this point. Then from the pheophytin, who is going to accept the electron? Plastoquinone is going to accept the electron. From the plastoquinone, who is going to see? Plastoquinone, upon accepting the electron, it is, listen, it is going to reduce it to PQH2. But friends, everyone listen my statement. If the plastoquinone wants to reduce it to PQH2, do you think electron is sufficient? No. Along with the electron, PQ can also require proton. Don't forget that point. Now from where the PQ is getting the proton? From the stroma. Very simple. From the stroma, I'll explain twice, listen. From the stroma, PQ accept the proton. From the pheophytin accept the electron. Then the PQ going to reduce it to PQH2. Now the PQH2, do you think the electron will be remain with the PQH2? No, not at all. The electron has passed to what? Cytochrome B6F. Then upon passing the electron, listen my friends, PQH2 oxidized back to PQ. Okay, electron sent here. What about the proton? Friends, the proton is going to released into the lumen. I will explain once again. This time will be clear to all of you. Listen carefully. What's happening? We are understanding how the ATP is basically synthesizing. I am explaining to all of you clearly that first P680 send the electron to pheophytin. Pheophytin to electron send to plastoquinone. Plastoquinone accept the electron from pheophytin, accept the proton from stroma, PQ reduced to PQH2. Then PQH2, it won't keep the electron with it. It is going to pass the electron to cytochrome B6F. So upon passing the electron to cytochrome B6F, PQH2 oxidized back to PQ. Electron it sent, no problem. Then what about the proton? The proton released into the lumen. Now what you can understand, like these protons are every time released into the lumen. And in the previous slide, I have explained it to all of you very clearly. When water molecule break down, what I told you, we are getting proton, we are getting electron, we are getting oxygen. What I have told to you friends, I told you very clearly that the protons are going to released into the lumen. Now friends, what's the point you have to understand? The concentration of protons in the lumen. What my friends? The concentration of protons in the lumens becoming extremely higher and higher and higher. Now when the concentration of proton becoming higher when compared to the stroma, so the protons start moving from lumen to the stroma via, how it will move, via an enzyme complex. What do we call that enzyme complex? We call it as F0 F1 complex. What do we call? F0 F1. The F0 F1 complex also call it as what? ATP synthase enzyme. My friends, we are reading from 9th and 10th standard. Always proton move in the form of what? Wave. Correct na? So friends, whenever the proton started moving in the form of a wave, it came out. Now in the lumen protons are high. Now it came to stroma. The charge got balanced. Now we all know very well, proton generates energy. Yes or no? Yes. So friends, proton generate the energy. That energy can be used. That energy which is generated by the proton, we will use to join the ADP with PI. Hence there is a formation of ATP. This is how the ATP can be synthesized. And how the force is coming? By the proton. That's why we call proton motive force. Okay, we call it as what? Proton motive force. Please kindly do not forget this particular point. So friends, this is about cyclic and non-cyclic. Now I have given a wonderful differences between the cyclic and non-cyclic. I'm requesting all of you to remember the differences. You will only commonly say, we read right, in the cyclic only one photosystem is there. Here both are there. Here P700 is the active reaction center. Why? Because chlorophyll A absorb the light maximum at 700 nanometer. Here also chlorophyll A absorb the light at 680 nanometer. And you remember, photolysis means the breakdown of water only takes place at non-cyclic, cannot takes place at cyclic. And we read, we called it as a cyclic photophosphorylation because electrons are always traveling in a cyclic manner. So next one, here electrons are traveling in a non-cyclic. In cyclic only ATP produced, reducing equivalent NADPH cannot produced. Here non-cyclic, both NADPH and ATP molecules are produced. And what is the another unique point you have to remember? Oxygen is not evolved as a byproduct. Of course true. Friends, water only not breaking down, then how come oxygen can be evolved? Oxygen cannot evolve. Here water is breaking, oxygen is going to evolve. Friends, I want every one of you without fail, please remember this particular inhibitors. In the upcoming slides, I'm going to explain you that. Cyclic photophosphor, see, because in the exam most commonly they use it to ask the questions about inhibitor. Cyclic photophosphorylation is affected by DCMU, dichlorophenyl dimethylurea. And non-cyclic photophosphorylation, it is affected by paraquat, which is called as methyl vialogen. Please keep that in your mind. Do not forget that. Okay, right now we completely understand the light reactions of photosynthesis except a little part. Before continuing that, let's solve some questions of the PYQ. Don't be hurry, think and answer. Okay, see here, in a photosynthetic electron transport, you can answer the questions very easily. Electron travels through carriers organized in the jet scheme. The following are indicated as directions of the electron flow. So friends, if you write When you get this kind of question, you just write in a paper. By that you don't get confused. We read, right? P700, how is the electron scheme? A naught, A1, then there is a series of iron sulfur X, iron sulfur A, iron sulfur B, then ferridoxin, then FNR, then NADP, and followed by NADPH. And when we read about the P680, what we discussed? P680, ferridoxin, plastoquinone A, plastoquinone B, cytochrome B6F, and plastocyanin, followed by P700. This is what we read in the previous. Now you only look into this. P680. So here after P680 pheophytin, right? Because they have asked us in the non-cyclic, not the cyclic. So P680 plastoquinone. This is wrong. Why it is wrong? Because after P680 the electron acceptor is the pheophytin. So pheophytin, quinone A, quinone B, cytochrome B6F, plastocyanin, P700. R is absolutely true. P700, after that A0, A1, iron sulfur X, A, B, FD, FNR, NADP. Absolutely true. Q is correct. R is also correct. Now if you look into the yes, P700, A1 they have given. No, absolutely wrong. Hence, my friends, Q and R is the correct answer. Then what is the correct answer? Here the correct answer will be the B. Do not forget that. Now look into this particular question. Which one of the following is the correct order of electron transport during the light reaction in the thylakoid membrane? Once again, same, right? You know this question, if you know the last electron acceptor is plastocyanin, and if you at least know this without looking into three options also, you can choose the right answer. Yes or no? P680 pheophytin they did not give. After the pheophytin it is what? Plastoquinone. After plastoquinone, cytochrome B6F. After cytochrome B6F, obviously friends, plastocyanin. Very simple point, right? So we will continue the topic. By that we will come back to solve the questions. As I told you, my friends, there are certain herbicides are there which are going to block the electron transport or electron flow. Friends, once again I am repeatedly saying to all of you, in our life sciences or biotechnology, in our exam, wherever you come across inhibitors, regulator, promoters, stimulators, we should always read and remember that, because in the exam these things most oftenly and repeatedly asked, and very easy also. So you look into this. The first one, DCMU, dichlorophenyl dimethyl urea. Sometimes instead of calling DCMU in the exam they will call diuron. Anything is same. What it is going to block, you only look into this DCMU. It is going to block the electron flow between what to what. You are able to see first P680, then send electrons to what? Pheophytin. Pheophytin to quinone A, quinone A to quinone B. Now you only look into this. Friends, this particular DCMU exactly block the electron flow by occupying the binding site located between the quinone A and quinone B. Friends, you only try to understand, if the electron is not moving from quinone A to quinone B, how does the electron flow will be continue? Then how can we produce the ATP and NADPH? Entire light reactions will be inhibited. It block the electron flow by occupying the binding site of plastoquinone, where normally plastoquinone B binds. Next, triazine, same, no doubt. Triazine also block electron flow by occupying the binding site of plastoquinone competitively, where quinone B binds. Same, DCMU and triazine having the same mechanism of action. Paraquat. So sometimes, my friends, in the exam, instead of giving the paraquat, they can also call it as a methyl vialogen. Anything is same. Where or what kind of action it can show? It accepts the electrons from early acceptors of PS1 and finally reacts with the O2 to form superoxide that damage specifically the lipid component of the chloroplast. Otherwise, you just keep it in your mind. Paraquat. You remember, friends, from P700, who is going to accept the electron? A naught. After A naught, A1. We read all this. From the P700, the paraquat come and block. What exactly? It will block the reaction center of P700. When paraquat block the reaction center of P700, do you think the P700 can release the electron? No, not at all. Even though it release the electron, no one can accept it. So whatever the electrons given by the P700, that is accepted by paraquat, and it will block entire electron transport flow. It activate several free radicals which can damage entire electron scheme or entire lipid components of the chloroplast, which inhibits entire light reactions of the photosynthesis. I'm requesting all of you without fail, read and remember the herbicides which are going to block the electron flow. Please kindly do not forget that point. Now next one. So you can look into this. The same question was asked here. A researcher wanted to study light reactions during photosynthesis by blocking photosynthetic electron flow using the herbicide DCMU and paraquat. The researcher listed the following observation. You can tell me easily, right? We just now read what the DCMU will do, what the paraquat will do. You know very well, DCMU occupy the binding site of plastoquinone. Methyl Viagen accept the electron from P700 and activate the free radicals. Now you only look into this. You can answer it very easily. Both DCMU and paraquat block the electron flow in photosystem 2. Wrong. DCMU in the photosystem 2. Both DCMU and paraquat block the electron flow in photosystem 1. No, not at all. DCMU block electron flow in photosystem 1. Paraquat block the electron flow in the photosystem 2. Now you only look into the picture. You can tell it easily. DCMU. What it is doing, friends? DCMU blocking the electron flow at the PS2. Paraquat at the PS1. Now what is the answer option they have given here? DCMU block the electron flow in photosystem 1. Wrong, right? It is blocking the electron flow in the photosystem 2. Paraquat in photosystem 1. Hence the correct option will be yes. DCMU block the electron flow in photosystem 1 while paraquat block the electron flow in the photosystem 1. So friends, such a simple questions we can expect in the exam. Okay. This is about the light reactions of photosynthesis. We are left with a little more part. So friends, that's all for this class, and in the next class we are going to discuss the left part of light reactions, then followed by the dark reaction. Because in the exam, if you miss the question from the light reaction, there are a great chances that we can expect the questions from the dark reaction from the C3 pathway, C4 pathway, then followed by, friends, CAM pathway. Yes or no? Yeah. Okay. I am requesting all of you, whomever did not watch the first part of the photosynthesis, watch it first and then you watch this. So in the next class of the photosynthesis, that is the final part, I will finish the dark reaction and photoprotective mechanisms of the plant. By that the entire photosynthesis will be completed. So friends, in order to download these notes, in the description I am going to give the link for this particular PDF or particular PPT. Okay. Thank you, friends. Thank you all. I'll see you in the next lecture. Thank you.